Okay, hello everyone. Apologies, we had a very odd technical glitch that cost us a few minutes there. Etay is now ready to give his talk. Etay, it looks like your screen share disappeared when we went
Yeah,
live for whatever reason.
I stopped it for a
Oh,
second.
perfect. Okay, good. That
Yeah.
wasn't another glitch. That's good. So
All
yes, so
right.
I will turn things over to Etay for his talk.
All right. Thank you. And yeah, I'll just quickly thank the organizers for this really interesting meeting. And I've in this session, as you saw, I structured some breaks, so that actually helps when there are some problems. But also, we've integrated question times for each talk. Obviously, my talk will go shorter, so I probably won't have time for questions, and I'll try to zip by so that it doesn't drag to the next slot. But if there is something that you really want to ask, please feel free. I will share my screen again. All right. Can you see it in full presentation mode?
I see the slides right now, not the presentation
Okay.
mode.
So it's not in full presentation mode?
Correct.
Okay. So I wonder. So it's not. You say that it's not, right, in full presentation?
Correct. It is not in presentation mode.
Okay.
I see the whole PowerPoint screen.
Okay. So you see the slides as well?
Yeah, I see the slides along the side.
Okay, cool. Let me just try one more time sharing the full screen, and if not, we'll just go forward as is. All right. One sec. All right. Is it full presentation mode now?
Let's see. It's taking a second to show up. There we go.
All
Perfect.
right. So I'll try to go faster. We've been doing in my lab studying depression using computational models of cortical circuits to try to understand the deficits in cortical processing and also the signatures in simulated EEG signals. So, sorry, yes. So the way we use models of human cortical microcircuits in mental health, we have detailed models that have the different cell types and detailed connectivity. And we study primarily depression, aging, and schizophrenia. Today I'll talk primarily about the depression studies we've been doing. And we have three central themes. One is studying the deficits in cortical processing. So, for example, extracting features of sensory stimuli. This kind of framework also translates to higher cognition as well. We also try to identify signatures of the changes, the cellular changes and circuit changes in depression, for example, in simulated EEG. That could serve as diagnostic biomarkers. And we also test new, very promising pharmacology for depression in silico. I will be talking about the first two projects today. So, depression or major depressive disorder, which is the longer term, but I'll call it depression for this presentation, is a leading cause of disability in Canada and worldwide. Obviously, you may have heard of serotonin drugs that have some success, but there are still a significant population of the patients that are treatment resistant. And in recent years, it seems that this treatment resistant depression is associated with a change in cortical inhibition. So in particular, there are interesting studies from CAMH that showed reduced SST expression in SST interneurons in depression. These are post-mortem studies from humans in healthy versus depression, showing about a 40% decrease across all layers. And we can use that kind of data to model changes in brain microcircuitry in depression. And the SST interneurons are a key type of interneuron that modulate firing in the circuit, especially during baseline. And that result is also supported by optogenetic studies in rodent, where they silence or activate SST, and they can either induce or alleviate symptoms of stress and depression. So that led to the hypothesis that if there is a reduced inhibition from SST interneurons, and that would mainly affect the baseline firing, then in depression it would be a higher firing, because less inhibition means that pyramidals can fire more. But because those SST interneurons do not play such a big role in response, then the effect on response in the circuit would be minimal. So if you look at response as signal and the baseline activity more as noise, this is the hypothesis that there is a reduced signal to noise ratio of cortical processing. And that could also explain the tendency and the symptoms in depression patients, where they're pretty much absorbed in inner loops, negative loops usually, and the symptom of rumination. So to study this and to test this hypothesis, we use computational models. Currently, it's pretty impossible to do in the living human brain. But because there is increasing data of neuronal properties and circuit properties, we can use it to constrain detailed models and study those questions. So I'll zoom over that part. Kent from my lab is going to give a trainee talk about it, so you can hear more of the details there. But I'll just say that we use data from collaborators in the Allen Brain Database to model pyramidal neurons and the three key interneuron types. We reproduce the firing pattern of each cell type and some dendritic properties, such as the SAG current that you heard of, that is a signature of H current. And this is all human data. Those are the PV and VIP, and you saw before the SST and pyramidal. We also constrain the models to reproduce synaptic properties according to previous human studies. There are nice studies about the connections in human tissue between pyramidal and SST, and also PV as well. And our models reproduce the synaptic amplitudes and properties. So after we had those components, we connected those building blocks into circuits. So we had the right dimensions of the circuit of layer 2-3 in human. We had the right cell proportions using human data from the Allen brain, and also connection probabilities between the different types in the circuit. And here the color coding will remain throughout the presentation. So the first step was to simulate baseline healthy activity. So we injected the circuit with a background excitatory input, and we tuned that input to allow the cells to fire at the rates that you see either in pyramidal in human, because we have that data from studies, or in the other cell types in rodents, because that data is not currently available in humans, and the model does that. So now that we had the model of a healthy cortical microcircuit, we applied the gene expression data from our collaborators at ChemH to model reduced inhibition in depression. And we applied it both for the synaptic inhibition and also the steady tonic inhibition that SST interneurons contribute to. And to model that, we used the recordings of tonic inhibition from our collaborators at the Valiant lab at UHM. And by the way, this project that I'm telling you about is submitted in its own bioarchive if you want to hear more about it. Right, so the next step was to compare the healthy and depression microcircuits in terms of baseline activity. So we simulated them, And in support of the hypothesis, we saw that depression microcircuit, as you see in purple, are about 50% more active in terms of pyramidal firing rate than the healthy microcircuits you see here in gray. And to understand how that also works in terms of the response, we modeled the response, because that was baseline that I just showed you, the response in the microcircuits according to neuron firing that you can record in vivo. Again, in rodents, because that data is not available in humans. But that kind of pattern and the involvement of different types is seen actually across species and across other areas such as visual cortex. So that's a pretty canonical response profile to a brief stimulus. So if the stimulus is presented at time 0, pyramidal neurons tend to be activated, followed by some steady recurrent activity. SST interneurons, as we said, are not so much playing a role, so their rate actually goes down, and that's also because they're inactivated by VIP and PV interneurons. So our models capture all of that, and that allowed us to compare the healthy and depression microcircuits. So as we saw before, there was an increase in baseline activity, but not so much of a change of response activity. So the overall signal-to-noise ratio went down in depression quite significantly. And the last piece of that project was to try to understand, okay, SNR is a pretty intuitive measure, but how does that really, how can we infer from this processing in the cortex? So we looked at the notion of stimulus detection. So if we plot the distribution of firing rates of pyramidals when there is a stimulus, the response, right, in the solid curve, and the baseline firing rates, we have two distributions that, according to decision theory, if you want to say whether there was a signal or not, you will put a decision boundary in the middle, the intersection, right? And as those curves become more overlapping, your inference would suffer from it. And you quantify it by the idea of failed and false detection, which are those two shaded areas, and that's the same idea as failed and false positives. False negatives and positives. So using this framework, we compared the rate of failed and false detection in the healthy, that you see here in gray, and the depression. And what we saw is a significant increase of false detection as well as failed detection in depression. So essentially, because there is a higher baseline firing and more noisy firing, that leads to a noisy processing that can be also measured by those measures. And nicely, that is sufficient to explain results that have been measured behaviorally in depression patients. So the part I want to give a little bit more detail on in the few minutes that are remaining is the follow-up work that we use this model for, that has been carried by Frank Mazza, and this is in preparation. So what we did, because we implemented the circuit in a physical platform called LFPI, that we actually put the different neurons in space, we can simulate, in addition to the spiking in the neurons, also the electrodes. We can measure LFP, we can measure EEG. That software accounts for the different media of the head that filter the signal. So we can try to identify signatures of the reduced SST inhibition in EEG. So the first nice thing that we saw is that an emergent property of that circuit was that it reproduces features of resting EEG that you see in humans. And that's an emergent property. We didn't try to fit it. It just, from the fitting all the synapses, neurons, and all the biophysics, we got that for free, and that's a nice validation. So the key properties that we saw and are in line with human EEG is a peak at the theta and alpha frequencies. Those are 4 to 12 hertz, and also a 1/f log relationship that you can see here on the log scale plot. And just to mention, this work is done with John Griffiths from our center. So another thing that is in line with real EEG is that this is not a continuous oscillation that you see. Here you see the frequencies on the y-axis over time, and you can see here it's more of an event that has a temporal signature in line with a certain band. So here you see a theta event, here you see an alpha and theta, here is another theta, and here is a bit of an alpha. So that's also very nice and in line with real EEG. So as you remember, we have also depression models. So we next sought to compare the EEG, the power spectral density plots, to see if there are any signatures or changes we can see in depression for the reduced SST inhibition. And what we see is an increase in power. And the question is whether it's a broadband or a more specific periodic increase. So what we did is separate the two components, the broadband and the periodic, and what we see is an increase in theta periodic power, which you can see here, and also a broadband increase in the various frequency bands. And those are essentially can serve as biomarkers for the reduced SST in depression. So to summarize, I showed you some data-driven models of human neuron synapses and microcircuits that we developed in the lab using and integrating a lot of new data that is out there in the literature and databases and collaborators. I showed you that we derived some depression microcircuit models using gene expression data from depression patients. And we used it to find two interesting things. First of all, what are the consequences on cortical processing? And we showed that there is an impaired signal detection due to a noisy activity in the circuit, a higher baseline activity. And we also find signatures for it in our simulation of the EEG. And we see a marked increase in theta and broadband power in depression. So lastly, I would like to thank the team. Alex and Kent carried the first project that I mentioned, and Frank carried the EEG project. I'd like to thank the different collaborators at UHN, KMH, and the Krembil Center for Neuroinformatics, and our very generous fundings that made it possible. So I think I'm actually on time.
Yeah, you rescued us, Etay. Thank you.
Yeah.
So we will. I guess we'll skip over to question period to get back on schedule here, if that's okay with you. And so I think Alex is getting set up in his session right now. So as soon as he's set up, he'll go live, and everyone will be drawn into that session as we've been doing. Apologies again for the delay with that technical hiccup, but thank you, Etay, for accommodating and helping us get back on track. So we'll see you all in the next session.
See you.